Right now, inside every single cell in your body, there is a message. Not a metaphor, not a philosophical concept, an actual biological message, written in a chemical code that your body has been copying and passing down for hundreds of thousands of years. And buried within that message, tucked inside sequences of nucleotides that most people will never think about, is a clue. A clue that points with remarkable consistency back to one place on Earth, Africa. Here's the question that sits at the heart of everything we're about to explore. Can scientists actually look at the DNA of a person alive today, someone sitting in London or Seoul or Sao Paulo or Lagos, and find within their genome detectable, measurable, undeniable evidence that their deepest ancestry traces back to Africa? Not through historical records, not through mythology or cultural tradition, but through the raw biological information written inside their cells. The answer, built from decades of genetic research, fossil analysis and migration studies, is yes. But that simple yes comes loaded with complexity, and the complexity is where things get genuinely fascinating, because what the evidence actually shows is both more precise and more nuanced than most popular accounts suggest. The headlines tend to flatten it. Humanity came from Africa, they say, which is true, but incomplete. The real story is richer, stranger, and more interesting than a single sentence can hold. Because here's the tension that runs through this entire subject. The science is real and the evidence is overwhelming, but it is also frequently oversimplified, sometimes misrepresented, and occasionally twisted into claims that go far beyond what the data actually supports. People hear that all humans share African origins, and they imagine a single moment, a single tribe, a single woman at the dawn of time. And while there are figures in genetics that have been given names like Eve, the reality is considerably more complicated than that image suggests. We'll get into exactly why. What we can say, with confidence grounded in actual evidence, is this. Every population on Earth carries within its genome patterns that geneticists can read like geological strata, layers of history deposited over time, mutations that accumulated at measurable rates, lineages that branched and diverged. And when you trace those lineages back far enough, following the branches toward their root, they converge in Africa. Not because scientists decided they should, but because that's where the oldest, most diverse, most branching genetic variation on Earth is found. The further a population is from Africa geographically, the less genetic diversity it tends to carry. That pattern has an explanation, and it's one of the most powerful arguments for African origins that exists. But none of this emerged from a single discovery or a single scientist's intuition. It came from an accumulation of evidence, genetics, fossils, ancient DNA recovered from bones tens of thousands of years old, isotope analysis, archaeological records of symbolic behavior and tool use. It came from researchers working across disciplines, sometimes agreeing, sometimes arguing fiercely, building a picture that is far more detailed today than it was even 20 years ago. Science doesn't hand you certainties here. It hands you probabilities, patterns, and occasionally surprises that force everyone to revise what they thought they knew. And that's what makes this worth understanding properly, rather than settling for the simplified version. Because the simplified version, satisfying as it is, misses the genuine wonder of what the evidence reveals. It misses the fact that ancient populations mixed in ways we're still mapping. It misses that some of the DNA inside modern humans came from lineages we only discovered in the last 15 years. It misses the open questions that scientists are still working through right now. So, over the next stretch of this journey, that's what we're going to explore. The genetic tools that made this research possible, the specific discoveries that built the case, the debates that still continue, and the distinction between what the science actually shows and what gets lost in translation between a research paper and a headline. Before we go further, though, I want to pause for a moment and ask you something. Where in the world are you listening from right now? What time is it where you are? Drop it in the comments. I always find it remarkable to think about how many different corners of this planet people are watching from. All of us tracing back to the same origin, separated now by oceans and time zones and languages, but connected by something written in every cell we carry. Tell me where you are. It's one of the quieter wonders of this kind of conversation, feeling the globe beneath all of us at once. Now, let's talk about what the science actually means. When scientists say humanity originated in Africa, they don't mean what most people initially picture. They're not saying that every person's entire ancestry remained in Africa until some recent historical moment. They're not saying that African populations today are somehow closer to a pure original form, while everyone else drifted away from it. And they're absolutely not saying that the journey from Africa to the rest of the world was a single event, a single group, a single departure. What they mean is something at once simpler and considerably more layered than any of those interpretations. Let's be precise about this, because precision is where the real story lives. The scientific consensus, built from converging lines of genetic and fossil evidence, is that anatomically modern humans, Homo sapiens, with the full skeletal architecture we recognize as our own, evolved in Africa. Not everywhere, simultaneously, not independently on multiple continents. In Africa. The oldest fossil evidence for our species comes from Africa. The deepest, most ancient branches of the human genetic tree are found in Africa. The greatest genetic diversity on the planet is concentrated in African populations, particularly among groups in sub-Saharan Africa who carry within their genomes a breadth of variation that dwarfs what you find anywhere else on Earth. These are not coincidences. They're signatures of origin. The model that captures all of this is called the Out of Africa model, sometimes referred to as the Recent African Origin model, and its core claim is that the ancestors of all living people outside Africa left that continent in one or more waves of migration, the most significant of which occurred roughly 50,000 to 70,000 years ago, possibly earlier. Small groups of Homo sapiens moved northward, crossed into the Levant, spread through Asia, eventually reached Europe, pushed into Australia, and tens of thousands of years after that, crossed into the Americas. As they moved, they diversified. Populations separated by geography accumulated different mutations, adapted to different climates, developed different physical characteristics. The branches of the human family tree spread across the globe. But here's where the story immediately gets more complicated, and this is something worth sitting with. Modern humans were not the first hominins to leave Africa. Not even close. If you've been following the earlier parts of this journey, you already know about Homo erectus, that tall, large-brained, fire-using species that walked out of Africa around 1.8 million years ago and spread across enormous stretches of Eurasia. They were in Georgia, in Java, in China, long before Homo sapiens existed as a species at all. Later, other archaic human populations were present in Europe and Asia, including Neanderthals in the west and a group we call the Denisovans in the east, whose existence we only confirmed from a tiny fragment of bone in a Siberian cave within the last 15 years. The world that our ancestors walked into when they left Africa was not empty. It was already inhabited by cousins, distant but real. That matters for what scientists mean by African origins, because the story of modern humans is not simply a story of migration into vacant landscapes. It's a story of encounter, and sometimes of intermingling. The populations that spread out of Africa met populations already living elsewhere, and the genetic evidence tells us with increasing clarity that they mixed. That complicates the clean picture, but doesn't undermine the central point. The trunk of the tree, the lineage from which all living humans descend, has its deepest roots in Africa. The branches crossed and tangled in ways we're still mapping, but trace any branch back far enough and it returns to that same ground. Now consider the timescales involved, because without a feel for the depths of time here, the whole picture blurs. When geneticists talk about the African origins of modern humans, they're primarily talking about events that happened hundreds of thousands of years ago. The emergence of anatomically modern humans in Africa is currently dated to at least 300,000 years ago, possibly earlier based on fossils found in Morocco at a site called Jebel Irhoud. The major migration out of Africa that seeded populations across the globe happened somewhere in the range of 50,000 to 70,000 years ago. Though some researchers argue for earlier dispersals that may have left limited, lasting genetic traces, these are not vague ancient times. These are specific, measurable intervals that geneticists can estimate by tracking the rate at which mutations accumulate in DNA over generations. Between those ancient departures and the world we live in today, an enormous amount has happened. Populations have split, diverged, collapsed, expanded, migrated again, and mixed with other populations repeatedly. Historical migrations, the kind recorded in written records, represent only the shallowest layer of a much deeper palimpsest. When you look at the genome of any living person today, you're looking at the accumulated result of all of it, ancient African origins layered under archaic admixture, layered under tens of thousands of years of regional diversification, layered under more recent historical movements. The recent stuff is easy to see. The ancient stuff requires more sophisticated tools to detect. But it's there, and this is perhaps the most important clarification of all. When scientists talk about all living people sharing deep common ancestry, they don't mean that we're all the same, or that ancestry is meaningless, or that recent history doesn't matter. They mean that if you follow any lineage backward through time, through the branching and mixing and diverging, you eventually arrive at a shared population in Africa, a population that was itself diverse, itself complex, itself not some simple primordial beginning, but an evolutionary chapter in a much longer story. The African origin of modern humans is not a poetic claim. It's a specific, testable, repeatedly tested scientific conclusion. And understanding what it does and doesn't claim is essential before we can understand what the DNA evidence actually shows us. So let's go there. Let's actually stand in Africa in deep time and look at what the fossil and archaeological record is telling us about where and how our species took shape. Because the picture that has emerged over the last few decades is not the one most people carry around in their heads. It's richer, more distributed, and in some ways more surprising. For most of the 20th century, the story of modern human origins had a pleasingly tidy geography. East Africa was the cradle. The Rift Valley was the nursery. Ethiopia, Kenya, Tanzania. These were the sacred sites, the places where the key specimens turned up, where the timelines seemed to converge. And there's genuine substance to that narrative. The East African Rift really is one of the most remarkable repositories of human evolutionary history on the planet. The geology there is cooperative, sediments layering in ways that preserve bones, volcanic ash, providing the datable horizons that let scientists assign ages with precision. But tidy geography has a way of reflecting the places scientists have been looking as much as the places where things actually happened. Start with Jebel Irhoud. It's a site in Morocco in northwestern Africa, nowhere near the East African Rift. Fossils were first found there in the 1960s, but for decades they were poorly dated and ambiguously interpreted. Then in 2017, a reanalysis using more sophisticated dating methods produced a result that genuinely reconfigured the field. The hominin remains from Jebel Irhoud were approximately 300,000 years old, and they showed features consistent with early Homo sapiens. Not fully modern in every detail, the face was somewhat more projecting, the brain case slightly more elongated than yours or mine, but within the range of what paleoanthropologists now recognize as the earliest expressions of our species, 300,000 years ago, in Morocco. That pushed the origin of Homo sapiens deeper in time than most models had assumed, and broader in geography than the East African story alone could accommodate. Then there's the Omo Kibish Formation in southwestern Ethiopia, where fossils designated Omo I and Omo II were excavated in the 1960s by Richard Leakey's team. Omo I in particular shows strikingly modern features: a high-domed cranium, a chin, a face that would not look entirely out of place in a contemporary population. Dating has been revised and debated repeatedly, but current estimates place these remains at around 195,000 years old, possibly older. And from Herto, also in Ethiopia, fossils dated to around 160,000 years ago show a morphology that some researchers describe as nearly fully modern, though with a few archaic characteristics that suggest the transformation was still in process, still a gradient rather than a switch being flipped. What emerges from these sites, and from other finds across the continent, is not the image of a single small founder population sitting in one valley, gradually becoming human and then spreading outward. What emerges instead is something more like a continental scale process. Multiple populations across Africa, separated by geography, shaped by different environments, evolving in partial isolation from one another, but never in complete isolation, exchanging genes when conditions allowed contact, diverging again when barriers reasserted themselves. The technical term researchers now use is African multi-regionalism, though that framing carries some baggage and means something quite different from the older multi-regional hypothesis that argued for independent evolution on separate continents. This is not that. This is one species taking shape across a large and varied continent, with different regional populations contributing to the eventual genome of modern Homo sapiens. And what was driving those patterns of separation and contact? Climate. Always climate, working through landscape. Africa, over the past 300,000 years, has not been a static backdrop. It has pulsed. The Sahara, which today seems like an absolute barrier between northern and sub-Saharan Africa, has repeatedly expanded and contracted. During wetter periods, what researchers call green Sahara phases, lakes formed, rivers ran, vegetation spread across what is now barren desert, and populations that had been separated could move and mix. During drier, colder periods, the Sahara returned. Forests retreated, grasslands shifted, and populations were again pushed apart, channeled along rivers, concentrated around refugia where water persisted. The coasts mattered too. Shellfish middens along the southern African coastline, particularly in places like Pinnacle Point and Blombos Cave, preserve evidence that populations there were exploiting marine resources with considerable sophistication. They were engraving ochre, making composite tools, using pigments in ways that suggest something more than pure survival calculus. These were people with complex behavior, living along a coastline that would have offered relatively stable resources even when interior conditions deteriorated. Coastal corridors along both the eastern and western edges of the continent may have served as highways for population movement during phases when interior Africa became less hospitable. The great river systems were another structuring force. The Congo, the Niger, the Nile, these are not just rivers. They are ecological corridors, population dividers, and occasionally population connectors, depending on which direction conditions were pushing people. A forest-adapted population on one side of a major river system might remain genetically distinct from a population on the other side for thousands of generations, accumulating different variants, and then come into contact during a wetter period when the forest expanded and the social boundaries became permeable. Here's why all of this matters for the genetic patterns we see today. When geneticists look at the diversity of living African populations, they find something that cannot be explained by a simple model of a single founding group and subsequent spread. The depth of genetic divergence between different African populations, particularly between groups like the San of southern Africa and populations in East or West Africa, reflects separations that go back hundreds of thousands of years. These are not recent differences. They are ancient. They are the signature of exactly the kind of long-term population structure, repeated separation, and partial contact that the fossil record is beginning to describe. The deep African diversity that geneticists keep returning to is not an accident or an anomaly. It is the accumulated record of a species spending most of its existence diversifying within one continent, shaped by a constantly shifting mosaic of grassland, forest, coast and desert, separated and reconnected by climate cycles that operated over tens of thousands of years. The populations that eventually left Africa carried with them only a fraction of that diversity, the genetic equivalent of a small book extracted from an enormous library. Africa retained the rest. And that library, those ancient lineages still present in African genomes today, is where some of the most extraordinary evidence for human origins is now being found. But to read it, scientists needed a new kind of tool. Not a fossil, not a stone tool, not a layer of volcanic ash. Something far smaller, far more precise, and far more personal. They needed to learn to read the molecule that carries your own history inside every cell of your body. Think about what is happening inside your body right now. In nearly every one of your roughly 37 trillion cells, there is a complete instruction manual for building and running a human being. 6 feet of it. If you uncoiled the whole thing, compressed into a space smaller than the period at the end of this sentence. That molecule is DNA, and for most of human history it sat there inaccessible, carrying its secrets in a language nobody could read. Then, in the second half of the 20th century, scientists began to crack the code. And what they found inside wasn't just biology, it was history. Compressed, layered, imperfect history, but history nonetheless. Here's the basic mechanism, and it's worth taking a moment with, because everything that follows depends on it. DNA is a sequence of chemical letters, 4 of them, abbreviated A, T, C, and G. Your genome contains roughly 3 billion of these letters arranged in a specific order. When cells divide, that sequence gets copied, and copying across 3 billion letters is not always perfect. Errors creep in. A letter gets substituted, a small section gets duplicated or deleted. Most of these errors happen in regions that don't do much, so they don't harm the organism. They just persist. They get passed down to offspring, who pass them down to their offspring, and so on through the generations. These accumulated errors are what geneticists call mutations, or, more neutrally, variants. And here is the crucial insight that turned DNA into a time machine. If 2 populations share a particular variant, they probably inherited it from a common ancestor who already carried it. The more variants 2 populations share, the more recently they diverged. The fewer they share, the longer ago their lineages separated. Relatedness leaves a signature in the genome, and if you can estimate how fast mutations accumulate, you can use the number of differences between 2 genomes to estimate how long ago their owners shared a common ancestor. That's the genetic clock. The idea sounds almost too elegant, and researchers are careful to acknowledge that it is imperfect in practice. Mutation rates are not perfectly constant. They vary between different regions of the genome, between different lineages, between males and females. Some mutations are invisible to natural selection and accumulate steadily, making them useful for timing. Others are subject to selection, meaning beneficial ones spread faster and harmful ones disappear faster than chance alone would predict, which throws off the clock. Population size matters too. Small populations lose variance through random chance, a process called genetic drift, which can make populations appear more distantly related than they actually are. Every estimate of divergence time comes with error bars, sometimes wide ones, and geneticists are genuinely humble about this. At least the good ones are. The clock is real, but it ticks unevenly. Despite those complications, the approach has been transformative, and it becomes even more powerful when you understand that not all DNA in your body is the same kind. Scientists distinguish between three main types of genetic material, and each tells a different part of the story. The first is mitochondrial DNA. Your mitochondria, the structures inside cells that generate energy, carry their own small, separate genome, entirely distinct from the main chromosomal DNA in the nucleus. Mitochondrial DNA is inherited almost exclusively through the maternal line. Your mitochondrial genome came from your mother, who got it from her mother, who got it from hers, in an unbroken chain of maternal descent, reaching back through time. Because it doesn't recombine, meaning it doesn't shuffle with a partner's DNA the way chromosomal DNA does, it accumulates mutations in a relatively clean, traceable way. Follow the mutations back far enough and you can, in principle, trace every living human's mitochondrial lineage to a single ancestral woman. We'll get to her shortly. The second is the Y-chromosome, which follows the opposite logic. Only biological males carry a Y-chromosome, and they inherit it from their fathers, who inherited it from their fathers, in an unbroken paternal chain. Like mitochondrial DNA, the Y-chromosome undergoes very little recombination, so its mutation record is relatively clean. It lets researchers trace paternal lineages backward through time with a similar kind of resolution. The third type is autosomal DNA, the remaining 22 pairs of chromosomes that both males and females carry. This is the vast majority of your genome, and it is shuffled and recombined every generation, meaning you inherit a mosaic from all your recent ancestors, not just your maternal or paternal line. Autosomal DNA is far more complex to analyze. But precisely because of that complexity, it carries far more information. It doesn't give you a single clean lineage running back in time. It gives you a sample of ancestry from across your entire family tree, hundreds, then thousands, then tens of thousands of ancestors, contributing fragments to the genome you carry today. Here is why this distinction matters enormously for the story of human origins. Mitochondrial DNA and the Y chromosome are powerful but narrow. Each traces exactly one line of descent through the labyrinth of your ancestry. If you go back 10 generations, you have over 1,000 ancestors, but your mitochondrial DNA represents only one of them, and your Y chromosome represents only one other. Using either alone to tell the story of a species is a bit like trying to describe a city by looking at a single street. You might learn something genuine. You might also get a very skewed impression. This is exactly why modern population genetics has moved decisively toward analyzing many genomes at once, comparing variation across whole populations rather than chasing single lineages. When researchers sequence the genomes of hundreds or thousands of people from different populations around the world and compare them, patterns emerge that no single marker could reveal on its own. The geography of genetic diversity becomes visible. The timing of population separations becomes estimable. The direction of ancient migrations becomes traceable. Old divergences that single marker studies missed begin to surface. The technical capacity to do this has grown at a pace that would have seemed like science fiction a generation ago. Sequencing the first human genome took over a decade and cost roughly $3 billion. Today, a full genome sequence costs less than a decent bicycle. The databases now contain hundreds of thousands of fully sequenced genomes from populations across every inhabited continent. Each one is a data point. Each one is a voice in a conversation that started around 300,000 years ago. And what those voices, taken together, are saying is remarkably consistent with what the fossils in Morocco and Ethiopia and South Africa have been suggesting. The deepest branches of the human family tree are all rooted in Africa. The oldest separations, the most ancient divergences between living populations, are found between groups within Africa, not between Africa and the rest of the world. The library analogy from the previous chapter holds up under genetic scrutiny. Africa kept the original manuscripts. Everyone else has copies of a subset. But before we map the whole library, let's look more closely at the two single-line stories, the mitochondrial and paternal trails, because they have their own drama. And the most famous figure in that drama is a woman who never knew she was famous. In 1987, a paper appeared in the journal Nature that set off a scientific and popular firestorm in roughly equal measure. The authors, Rebecca Cann, Mark Stoneking and Alan Wilson, had collected mitochondrial DNA samples from 147 people representing populations from Africa, Asia, Europe, Australia, and New Guinea. They analysed the differences between those samples, built a family tree of mitochondrial lineages, and followed the branches backward to a single point of convergence. That point, they argued, represented a woman who lived in Africa, probably sub-Saharan Africa, somewhere between 140,000 and 200,000 years ago. The press got hold of it and gave her a name, Mitochondrial Eve. The name was irresistible and immediately misleading. Within weeks, the story had been absorbed into popular culture in a form that bore only a faint resemblance to what the scientists actually claimed. Before we talk about what the finding meant, it's worth spending a moment on why mitochondrial DNA attracted so much attention in the first place. Because it wasn't an arbitrary choice. Mitochondrial DNA has several properties that made it unusually appealing to early geneticists trying to trace human ancestry. It's small, only about 16,500 base pairs compared to the 3 billion in your nuclear genome, which made it far easier to sequence with the technology available in the 1980s. It's present in cells in high copy numbers because each cell contains many mitochondria, so there's more of it to work with, even in degraded samples. It mutates at a reasonably steady and relatively fast rate compared to much of the nuclear genome, which gives it good resolution for tracking events in the recent evolutionary past, the last few 100,000 years. And crucially, as the previous section established, it doesn't recombine. It passes from mother to child essentially intact, accumulating mutations cleanly along the maternal line, without the scrambling effect you get with chromosomal DNA, that makes it comparatively straightforward to build a lineage tree. You're following one thread, not trying to untangle a tapestry. So when Cann, Stoneking, and Wilson traced all living mitochondrial lineages backward and found that they converged on a single ancestral sequence, that convergence was real. There genuinely is, according to everything we know from genetics, a single woman whose mitochondrial DNA is the direct maternal ancestor of every mitochondrial genome carried by every person alive today. That is not a metaphor. That is a mathematical and genealogical fact. But here is what it doesn't mean, and this is where popular understanding went badly wrong. Mitochondrial Eve was not the first woman. She was not Adam's companion in any biological sense. She was not the only woman alive when she lived. She was not even the most important woman of her era in any meaningful evolutionary sense. She was simply the most recent woman from whom an unbroken maternal line connects to every living human being. That qualifier, unbroken maternal line, is doing enormous work in that sentence, and it almost never made it into the newspaper headlines. Think about what it actually takes for a maternal line to go extinct. All you need is for one woman in any generation to have no daughters. Sons can carry her mitochondrial DNA in their bodies, but they can't pass it on. So every time a woman's lineage produces only male children, or no surviving children at all, that entire mitochondrial line disappears from future generations. And this was happening constantly, across all populations, throughout all of human prehistory. Maternal lines were going extinct not because those women were dying without children, but because of the ordinary randomness of which sex children happened to be born. Generation by generation, the number of distinct maternal lineages shrank, not because of any dramatic event, but because chance keeps pruning the tree. Follow that process back far enough, and eventually you arrive at the last common ancestral node. That node is mitochondrial Eve. At the time she lived, there were thousands of other women alive. Some of them were her relatives. Some may have been more influential in a social or reproductive sense. Their nuclear DNA, the bulk of their genetic contribution, is almost certainly present in living humans today, mixed and distributed invisibly across genomes on every continent. But their mitochondrial lines happened to go extinct at some point between then and now. Hers didn't. That's the whole story. She won a particular game of genealogical roulette that her contemporaries eventually lost, not because she was special, but because the thread from her to the present remained unbroken through sheer persistence. The inference that she lived in Africa came from the structure of the family tree itself. When the researchers organized all 147 mitochondrial sequences by similarity, the deepest branches, the most ancient divergences, were found among populations of African descent. The tree had its deepest roots there. If you want to find where a tree started growing, you look at where the oldest branches split from the trunk. Those oldest branches were African. It was a striking confirmation, from an entirely independent line of evidence, of what the fossils were already suggesting about the depth of African human ancestry. But the finding also came with complications that the initial wave of press coverage largely ignored. The statistical methods used to build the tree were challenged by other researchers, who pointed out that the same data could produce multiple different tree structures, not all of them rooting in Africa. Later analyses with improved methods and larger sample sets supported the African root more robustly, but the early confidence of the 1987 paper was somewhat overstated. Science, in other words, did what science is supposed to do. It argued with itself. It refined the claim. The African origin of the mitochondrial lineage survived that scrutiny, but not without earning it. The bigger problem was what the popular story did with the word Eve. It loaded an essentially statistical finding with narrative weight it couldn't support. Mitochondrial Eve became a character, a single woman who was the mother of humanity, a biological stand-in for a creation story. That framing made the finding seem more dramatic, more singular, more destined than it actually was. And it set up a misunderstanding that lingers to this day, the idea that human origins can be traced to one person, one moment, one dramatic point of emergence. Real ancestry doesn't work that way. You are not descended from one woman. You are descended from thousands of women who were alive when mitochondrial Eve walked the African landscape, and their contributions to your genome are just as real as hers. She is simply the one whose mitochondrial signature happened to survive the long lottery of maternal inheritance. She is a marker, not a mother of mankind, a signpost pointing toward Africa, not a door through which all of humanity passed. She is remarkable enough as what she actually is. She doesn't need to be more. If mitochondrial DNA is the mother's whisper through time, the Y-chromosome is the father's echo, where mitochondria trace a single, unbroken maternal thread. The Y-chromosome does something similar for one very particular slice of the male line. With one major caveat: only people who are biologically male, and specifically those with a Y-chromosome, carry it. A father passes his Y to his sons. His daughters don't receive it at all. Those sons, if they have sons, pass that same Y onward, generation after generation, with only occasional changes. No recombination with a partner's Y, no mixing the way your other chromosomes mix. It's a mostly intact baton, handed from father to son down the centuries. Geneticists realized that this made the Y a kind of counterpart to mitochondrial DNA. If mitochondrial genomes let you follow one narrow maternal thread, Perhaps the Y chromosome could let you follow a narrow paternal one. So, in the 1990s and early 2000s, as sequencing technology improved, researchers began cataloguing tiny variations on the Y, single-letter changes called SNPs, short repeated sequences called microsatellites. They used those differences to build a branching tree of Y chromosome lineages, what geneticists call haplogroups. When they followed those branches backward, through all the living Y chromosomes they had sampled, they found something strikingly familiar: convergence. Just as all mitochondrial lines meet at a common ancestor in the past, all known Y chromosomes trace back to a single ancestral Y carried by one man. He has been given a name every bit as dramatic and every bit as misleading as mitochondrial Eve's Y-Chromosomal Adam. Once again, the language plays tricks on the mind. Y-Chromosomal Adam was not the first man. He was not the only man alive in his time. He was not the biological partner of mitochondrial Eve. In fact, the best estimates suggest they lived tens of thousands of years apart, perhaps separated by more than 100,000 years. There was never a literal couple walking some primeval African landscape, destined to become the exclusive ancestors of all of us. That image is a storybook overlay, not a reflection of the data. What Y-chromosomal Adam represents is conceptually identical to what mitochondrial Eve represents, the most recent individual from whom all living carriers of a particular unbroken line inherit that line. In his case, the line is father to son, Y chromosome to Y chromosome. At the time he lived, there were many other men whose nuclear DNA spreads into the present just as widely. They simply happened by the usual roll of genealogical dice, to leave no surviving, uninterrupted chains of sons that carry their original Y. Imagine a group of related families. One man has 3 sons and several grandsons, but at some point down the generations, his male line produces only daughters. Another man's line does the same. Over time, most male lines in this strict Y chromosome sense wink out in this way. Lines can also disappear in droughts, migrations, wars, or just bad luck. Follow this winnowing process back long enough, and eventually all surviving Y lineages collapse to one ancestor whose male line never quite broke. That man is Y chromosomal Adam. Not a patriarch chosen by nature for greatness, just someone whose Y chromosome luck held. Even though the concept is narrow, the Y chromosome has turned out to be a powerful tool for tracing how different male lineages spread and branched as humans moved across the globe. Those variations geneticists catalogued, the SNPs and repeats, tend to cluster into distinct patterns. Each pattern defines a haplogroup, a clan of Y chromosomes that share a common ancestor marked by a particular mutation. Some of those haplogroups are ancient and mostly African. Others are younger and are found mainly outside Africa. For example, one major Y lineage, usually labelled haplogroup A, is found almost entirely in certain African populations and sits very close to the base of the Y chromosome tree. Other early branching haplogroups like B are also predominantly African. The branches that come later, C, D, F, and the many sub-branches, such as R and O, that dominate in Europe and much of Asia, emerge further up the tree, downstream of those older African splits. When you map where these Y haplogroups appear around the world, you see a pattern that mirrors in broad strokes what fossils and mitochondrial DNA already suggested. The deepest, most divergent Y lineages are in Africa. The branches that dominate outside Africa trace back to a subset of African Y chromosomes that left the continent relatively recently on an evolutionary timescale. It's the same story the mitochondrial tree was telling, now retold in the language of the paternal line. But if the Y chromosome reinforces the African origin of modern humans, it also underlines a crucial limitation. Just as mitochondrial DNA is only one very thin thread of ancestry, The Y chromosome is another. Together they account for 2 single lines out of the thousands of genealogical paths that lead to you. Your father's father's father, and so on in one unbroken chain. Your mother's mother's mother in another. That's it. Most of your genome has nothing to do with either of those special lines. It comes from people whose mitochondrial and Y lineages went extinct long ago, but whose autosomal DNA, the DNA on the other chromosomes, has been mixing and remixing for hundreds of thousands of years. By focusing on Y and mitochondrial DNA, we are staring at 2 razor-thin threads and trying to infer the shape of an entire tapestry. Still, those threads are useful precisely because they are simple. They don't recombine, so their branching patterns are easier to read. And the fact that both of them, read independently, point their deepest roots toward Africa is not trivial. Mitochondrial Eve's lineage rooted in Africa. Y-Chromosomal Adam's lineage rooted in Africa. The oldest splits in both trees are among African populations. When 2 separate, unlinked genetic systems converge on the same geographic story, and that story matches what the fossils and archaeology have been hinting at for decades, the case for a deep African origin of our species gets stronger. The Y chromosome doesn't tell the whole story. It tells a story about fathers and sons, about how a few male lineages spread and others disappeared. But when you place that narrow paternal trail alongside the maternal one, and then alongside the broader river of the rest of our DNA, a coherent picture starts to emerge. To see that bigger picture, though, scientists needed a different kind of tool, one that could look beyond these special lineages and read history in the shifting patterns of the entire genome. That's where DNA truly became a time machine. When scientists first began using that time machine on whole genomes, a pattern emerged, like a faint watermark seeping through paper. Different methods, different datasets, different teams, and yet the outlines kept lining up. At some point in the late Pleistocene, a group of Homo sapiens left Africa and their descendants spread across the rest of the world. Not the first humans to leave the continent, remember. Homo erectus had blazed that trail over 1 million years earlier. But this time it was different. This time it was us. The timing comes into focus when you layer genetics on top of archaeology. In Israel, at sites like Misliya, there are fossils of early Homo sapiens more than 170,000 years old, suggesting that our species had already probed beyond Africa long before the main dispersal. In Greece, and possibly China, there are tantalizing hints of even earlier excursions. These may represent failed or partial expansions, populations that did not end up contributing much, if anything, to living people. Your genome, though, is clearest about a later wave. Studies that compare the genetic variation of people from different continents converge on a window, roughly 50,000 to 70,000 years ago, for a major, successful dispersal of modern humans out of Africa. Within that range, estimates wobble depending on assumptions, mutation rates, and the particular populations sampled. But you keep returning to that broad slice of time. How do we know? One line of reasoning comes from the amount of genetic diversity found inside and outside Africa. Imagine a large ancient population in Africa, accumulating mutations over hundreds of thousands of years. Now, imagine that a relatively small subset of that population leaves. Those migrants carry with them only a fraction of the total genetic variation present at home. Over time, their descendants diversify too, but their starting palette is restricted. When you compare modern genomes, you see exactly that pattern. African populations today harbour more genetic variation overall. Non-African populations share a subset of that variation, plus a smaller number of newer region-specific changes. This is the signature of what geneticists call a founder effect. Whenever a small group branches off from a larger population and becomes isolated, chance plays an outsized role in which genetic variants make the jump and which are left behind. It's like scooping a cup of water from a vast swirling lake. In that cup, some dissolved molecules of genetic variation may be very common, others absent entirely, just by luck. If that cup of water is then poured into new ponds, and those ponds evaporate and refill and branch, the original skew gets amplified. The rare variants that happen to be overrepresented in the founding group may become common across a whole region of the world. Variants that were common in the original lake but didn't happen to be in the cup may be nearly absent outside Africa. The smaller the founding group, the stronger this effect. This is why a relatively small number of people leaving Africa could leave such a strong imprint on the genomes of billions. You don't need a vast caravan of migrants, just a population big enough to be viable, small enough to act as a genetic bottleneck, and persistent enough to grow and spread over thousands of generations. Your DNA still carries that bottleneck's echo compared to the deep, branching forest of variation within Africa. The rest of humanity's genome looks like a handful of saplings pruned from that forest and planted abroad. But this doesn't mean there was a single dramatic exodus, one tribe marching north and fanning out across Eurasia in a neat, orderly wave. The picture that emerges from both DNA and archaeology is more tangled, more human. There were likely multiple routes, multiple pulses, expansions and contractions tied to climate, sea level and opportunity. One obvious corridor leads through northeast Africa into the Sinai and Levant. When climate conditions were favourable, this region could be green and productive, a bridge rather than a barrier. Fossil and stone tool sites there show a long, complex history of human presence, including both our species and other hominins like Neanderthals. It's easy to imagine groups moving back and forth along this land route over tens of thousands of years, sometimes pushed out by drought, sometimes drawn in by rains. Another set of possibilities lies to the south. During periods of lower sea level, the Bab el-Mandeb Strait between the Horn of Africa and the Arabian Peninsula narrowed. Perhaps people used simple watercraft. Perhaps they took advantage of exposed islands and shoals. Once in Arabia, coastal corridors along the shores of the Red Sea, the Arabian Sea, and the Indian Ocean could have provided a chain of habitable niches, shellfish, mangroves, estuaries rich in life. Some models suggest that relatively rapid dispersals along these southern coasts might explain how Homo sapiens reached Southeast Asia and Australia by at least 50,000 to 65,000 years ago. These routes are not mutually exclusive. There may have been an early trickle through the Levant, a later surge along the southern shore, back migration into Africa, populations that advanced and then retreated as deserts expanded and contracted. Genetic data even hint that different non-African populations may descend from slightly different mixtures of early exiting groups, all ultimately drawn from African ancestors, but shaped by distinct histories of isolation and contact. When you fold all this complexity back into that simple observation, less genetic diversity outside Africa, more within, the story sharpens. The major non-African lineages, whether you track them with mitochondrial DNA, Y-chromosomes, or the full genome, branch off from within the African tree. They do not form a separate, older trunk. They are twigs from African branches that pruned themselves through those founder effects and bottlenecks as they moved into new lands. This is why, if you randomly sample people in Europe, Asia, the Americas, or Oceania, the variation you see almost always nests inside the broader variation sampled across African populations. Non-African genomes are not impoverished in any simple sense. They've had tens of thousands of years to develop their own unique combinations and adaptations, but they began as a subset. Your DNA, if you are of largely non-African ancestry, still carries that narrowing. It remembers in the reduced spread of its variants that your branch of the family tree passed through a tight gateway somewhere near the northeastern corner of Africa or along its southern shores. And if you are of African ancestry, especially from populations that remained within the continent for most of their history, your DNA holds an even older memory. It reflects not just that outward journey, but the deep, branching complexity that existed long before anyone stepped beyond Africa's borders. So why is that inner forest of variation in Africa so much richer, so much more tangled than the groves we see elsewhere? What is your DNA actually remembering when it points back again and again to that continent? Part of the answer is time. Not the dramatic cinematic kind with big migrations and sudden inventions, but the slow grinding accumulation of generations. Modern Homo sapiens, as a distinct species, almost certainly evolved in Africa. The earliest fossils that most researchers agree look like us in an anatomical sense come from places like Jebel Irhoud in Morocco, Omo Kibish and Herto in Ethiopia, with ages reaching back roughly 300,000 years, and perhaps a bit more. Those early people weren't living in one unified Eden. They were scattered across different regions and habitats, highlands and lowlands, coasts and interior basins, forests, grasslands, and everything in between. Each of those environments is a stage with its own script. Different food sources, different pathogens, different predators, different climate rhythms. Put populations in those settings for tens or hundreds of thousands of years. Allow them to expand and contract with wet phases and dry phases, and the genome responds. Mutations arise by chance. Some are weeded out, some drift, some are favoured. Lineages split, reconnect, overlap. Over deep time, that becomes a web of variation. You can think of this as Africa hosting not one ancestral population, but many partially connected ones. Geneticists sometimes talk about population structure, the idea that groups can be separate enough for their gene pools to diverge, but not so isolated that they never exchange genes. Africa's long-term structure looks more like a braided river than a single channel. Streams separate, flow apart for a while, then rejoin. Each branch picks up its own silt, its own distinctive variants, before mixing again. Now contrast that with what happened when some of those African lineages left. When a subset of people walked out of Africa 50,000 to 70,000 years ago, they carried only a slice of the standing diversity with them. That was the founder effect we just explored. But that first narrowing wasn't the last. As those migrants pushed into new terrain, their descendants repeatedly passed through bottlenecks. A bottleneck is simply a sharp reduction in population size. It might be caused by climate shifts, disease, competition with other hominins, unlucky geography. Whatever the cause, when numbers crash, many genetic variants crash out of existence with them, purely by chance. When the population later rebounds, it does so from a smaller set of surviving variants. Leaving Africa, bottleneck. Crossing a difficult corridor like the Arabian deserts or the Himalayas, bottleneck. A small band of people making it into a new island, a new valley, a new continent like the Americas. More bottlenecks. Imagine copying a book, then copying the copy, then copying that copy onto thinner and thinner paper, occasionally tearing out a few pages and rewriting them from memory. You will still end up with a coherent story in the end, but some phrases, some paragraphs, some marginal scribbles from the original will be gone. Outside Africa, the story of our species has been recopied through multiple sequential bottlenecks. Inside Africa, the original library was never completely closed. This is why genetic surveys consistently show that two individuals chosen at random from different African populations are, on average, more genetically different from each other than two individuals chosen from different non-African continents. A hunter-gatherer from southern Africa and a farmer from West Africa may carry more differences between them than a person from China and a person from Peru. That can feel counterintuitive if you grew up thinking in continental categories. But the genome doesn't respect those political lines. It remembers history, not maps. Crucially, that higher diversity doesn't mean more evolved or better. Evolution is not a ladder with some populations higher than others. It's a branching process. More diversity simply means more branches have persisted, more twigs have been allowed to grow, fewer have been pruned away by bottlenecks. Think of it like this. If Africa is the old, sprawling city where humanity has lived the longest, non-African regions are newer suburbs built by people who moved out. The suburbs have their own innovations, their own street layouts, their own local cultures. But if you want to see the deepest layering of human history, the oldest alleys, the overlapping neighbourhoods, the districts that rose and fell and rose again, you go back to the city. Africa's deep diversity is also shaped by its demographic scale. Over much of our species' history, the total number of people living in Africa was probably larger than the total number living elsewhere. More people means more births. More births mean more opportunities for new mutations. Most of those changes are neutral. Some are harmful and get weeded out. A few are useful and spread. Over hundreds of thousands of years, that steady churn adds up. On top of that, Africa's environmental mosaic never stopped shifting. Sahara deserts blooming into grassland and then drying again. Forest belts advancing and retreating. Rift valleys opening, lakes filling and shrinking. Each shift reshuffled populations, nudging groups together and apart, sometimes isolating them long enough to carve out distinct genetic signatures, then bringing them back into contact so those signatures could blend. When researchers model all this, population size, structure, mutation rates, bottlenecks, migrations, and then compare the results to real genetic data, the scenario that best fits is one where modern humans have their deepest, longest history within Africa. Non-African lineages are always younger branches, always subsets, no matter which part of the genome you study. Mitochondrial DNA, inherited through the maternal line, shows its greatest variation in African populations. Y-Chromosomes, tracing paternal lineages, do the same. Whole genome analyses that ignore any single family line and look at the entire chromosomal tapestry converge on the same pattern. The tree keeps rooting deepest in Africa, with the richest tangle of branches, and the major non-African lineages consistently emerging from within that tangle rather than standing apart from it. This is why geneticists treat Africa's diversity as one of the strongest lines of evidence for an African origin of modern humans. It isn't just that African populations are different from the rest of the world. It's that the rest of the world looks genomically like a recent sampling from within Africa's older, broader variation. Your DNA, whether you know your ancestry or not, carries that asymmetric history. If your heritage is largely non-African, it bears the marks of those bottlenecks and founder events that narrowed a once immense range of possibilities into the set your ancestors carried abroad. If your heritage is African, it holds echoes of a much longer, more intricate demographic story, one in which your line wove in and out of many others long before the first footprints were left in the sands of Arabia or the steppes of Eurasia. And yet even this is only part of the memory. Because your genome doesn't just remember Africa as a place where our species began. It also remembers the other humans we met along the way, cousins from different branches of the hominin family tree, whose DNA still lingers, faint but detectable inside you. So far we've followed two very particular threads through that story: mitochondria on the maternal side, Y-chromosomes on the paternal. They're powerful, they're elegant, but they're also narrow. One line up the family tree, one ancestor per generation. Real ancestry is messier than that. If you go back 10 generations, you don't just have one ancestral woman and one ancestral man. You have up to 1,024 ancestors at that level alone. 20 generations back, you're into the millions. Of course, Those numbers overlap in reality, because cousins marry cousins and lines fold back on themselves. But the basic point stands: your family tree isn't a straight trunk with two neat roots. It's a thicket. Mitochondrial DNA and the Y chromosome are like single paths threading up through that thicket, useful for seeing certain patterns, useless for others. Your mother's 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 line might all be from one region, while all your other ancestors from that same region, on different branches, are completely invisible to mtDNA. The same for the Y. Change a single father in your deep past, and that entire paternal story flips, even though the vast majority of your ancestors stay the same. To grasp what your DNA really remembers, you have to step away from those single lines and look at the whole forest. That's what autosomal DNA does. Everything that isn't mtDNA or the Y chromosome sits on your autosomes, the numbered chromosomes you inherit from both parents. Half your autosomal genome came from your mother, half from your father. They, in turn, got halves from their parents, and so on. With each generation, the genome is cut, shuffled, and reassembled. Segments from thousands of different ancestors end up stitched together into the string of bases that is you. Think of your autosomal DNA as a mosaic made from countless tiny tiles. Each tile is a stretch of sequence that once belonged, intact, to some ancestor in your past. Over time, recombination breaks those stretches into smaller and smaller pieces. The further back you go, the fewer tiles any single ancestor contributes, and the smaller those tiles become. Eventually, beyond a certain number of generations, the chance that you carry a sizable piece from any particular person dwindles towards zero. That doesn't mean the pattern disappears, it just changes scale. Zoomed in, the mosaic looks like noise, millions of positions where you might have one variant or another. But zoomed out across whole genomes and whole populations, regularities emerge. Certain combinations of variants tend to travel together. Certain arrangements of tiles show up more often in some groups than in others. These aren't markers of purity or fixed categories, they're signatures of shared history. Genome-wide studies work by reading those signatures across thousands, sometimes hundreds of thousands of people. Instead of asking, "Do you share this one maternal ancestor?" they ask, "How many segments, how many patterns do your genomes have in common? How big are those shared segments? How are they arranged? How do they compare to the segments in other populations?" It turns out that if you take two random people anywhere on Earth and you look across their autosomes, you will find shared chunks of DNA. Some are large and easy to spot, reflecting relatively recent common ancestry. Great-grandparents, cousins, populations that have mixed in the last few hundred or few thousand years. Others are tiny, almost lost in the background, reflecting connections tens of thousands of years old. Now scale up. Look not at two genomes, but at thousands spread around the globe. Feed that information into statistical models that simulate different demographic histories. Populations splitting, shrinking, expanding, mixing back together. You can then ask a question that sounds philosophical but is in fact mathematical. What sequence of events, what branching and blending would most likely produce the pattern of shared segments we see today? The answer over and over points back to Africa. In these models, non-African populations consistently emerge as offshoots from within a broader, older African diversity. Their genomes are best explained as having passed through a relatively recent bottleneck, followed by expansion and subsequent mixtures with each other, and with archaic hominins like Neanderthals and Denisovans. African populations, by contrast, are best explained as having maintained larger effective population sizes for longer, with multiple semi-isolated groups exchanging genes over deep time. You can see echoes of that history in the lengths of shared segments. a long, unbroken stretch of identical DNA between two people usually points to a recent common ancestor. Shorter, more fragmented stretches suggest that the shared ancestor lived further back, with more recombination chopping their contribution into pieces. When researchers map the distribution of those stretch lengths across populations, they can infer not only that African groups hold more variation overall, but also that many of their shared ancestries reach deeper into time than comparable connections outside Africa. There are also subtler patterns, certain configurations of rare variants, their frequencies and how they co-occur, carry the imprint of ancient population splits. When a once-connected group divides and stays mostly separate for a long while, mutations that arise in one branch don't appear in the other. Later, if those branches mix again, you get a telltale blend. Some regions of the genome look more like one ancestral population, some more like another. By teasing apart these mixtures, scientists can reconstruct episodes where African groups diverged, adapted in different environments, and then reconnected long before any ancestors of today's non-Africans left the continent. In this way, autosomal DNA is less like a family tree and more like a palimpsest, a manuscript written over many times. Earlier chapters are faint, overwritten by more recent events, but they're not completely erased. With the right tools, with enough genomes, you can make out the older writing beneath the newer script, and when you do, the underlying story is consistent. Whether you trace single lines like mtDNA and Y-chromosomes, or you let the entire genome speak at once, the deepest, most persistent signals of origin run through Africa. Every living human anywhere in the world carries autosomal segments that tie them back to populations that lived there. The size and arrangement of those segments, the way they cluster and diverge, fit best with a past in which our species' longest, most intricate history unfolded on that continent, and only later branched out across the rest of the globe. If there's any sense in which your DNA remembers Africa beyond poetry or metaphor, this is it. Not one mythical mother or one founding father, but a vast ensemble of ancestors, their genetic traces scattered and recombined across your chromosomes. The memory isn't a single voice singing a solo, it's a chorus, and the strongest, oldest harmonies in that chorus still carry the resonance of Africa. For a long time, that chorus of living genomes was all we had to listen to. Every model of human history, every timeline of migrations and splits, came from reading patterns in the DNA of people alive today and extrapolating backward. It's a bit like walking into a crowded concert hall after the music stops and trying to guess what symphony was just played, based only on the lingering echoes. Then very recently, something extraordinary happened. Scientists learned how to coax DNA out of the dead. Not metaphorically, not in some mystical sense, but literally. Powdered bone, flakes of tooth enamel, tiny fragments of skull, pulled from caves and burial sites. For decades, those fossils could tell us about shape, size, sometimes cause of death, sometimes diet. But they were mute about genetics. DNA is a fragile molecule. It breaks, it cross-links, it's chewed up by microbes, dissolved by water, shredded by heat. In most climates, after a few tens of thousands of years, the record is gone. The breakthrough came from a combination of clean room techniques, chemistry, and brute force sequencing. Researchers learned how to work in sealed labs, where a single dropped skin cell from a modern technician wouldn't swamp the ancient signal. They learned how to fish out the few surviving fragments of ancient DNA from a sea of microbial contamination. They built machines that could read billions of short, damaged pieces and computational tools that could stitch those pieces back into something recognizable. The first truly high-quality ancient human genomes, from Neanderthal bones and a small girl's finger bone found in Denisova Cave in Siberia, were a revelation. For the first time, We weren't just guessing what the ancestral symphony sounded like. We were dropping the needle on an old recording. But there's a catch, and it matters for Africa. Ancient DNA survives best in cool, stable, relatively dry environments. Caves in Siberia. High-altitude plateaus, permafrost. The very places where, for most of our species' history, almost nobody lived. The regions that matter most for our origin story, tropical and subtropical Africa, are brutal on DNA. Heat speeds up chemical reactions that break bonds. Moisture seeps into bone and carries away genetic material. Microbes thrive and eat whatever is left. So the continent that holds the deepest history of our species is, paradoxically, the hardest place from which to recover that history at the molecular level. That doesn't mean there's nothing. It means the record is patchy, hard-won and recent. A handful of genomes from ancient individuals in places like Ethiopia, Malawi, South Africa, Morocco are beginning to come online, each one the product of years of work and often only partial in coverage, compared to the hundreds of high-quality ancient genomes from Europe and parts of Asia. Africa is still vastly underrepresented. Yet even this imbalance has value, because genomes from ancient Eurasian humans can still tell us a great deal about what must have been happening in Africa before anyone left. Remember those models based solely on living people, the ones that said, an African population, or a network of populations, gives rise to a subset that moves out into Eurasia, experiences a bottleneck, then expands and mixes with Neanderthals and Denisovans. Ancient DNA lets us test that picture directly. Take a hunter-gatherer from Ice Age Europe, a 45,000-year-old bone from a cave in Romania. Extract the DNA, sequence it, and suddenly you can see, base by base, how this person relates to both modern Eurasians and modern Africans. You can measure exactly how much Neanderthal ancestry they carried, where in their genome those archaic segments were located, how long those segments were. Those segment lengths, in turn, translate into a time frame for when Neanderthals and the ancestors of modern humans interbred. Do the same for another individual, 10,000 years later, in a different part of Europe, or for early farmers in Anatolia, or ancient foragers in Siberia, and a pattern emerges. You can literally watch ancestries waxing and waning across time. You can see when a new population arrives in a region because, overnight in archaeological terms, the genetic profile of the burials changes. Those shifts confirm key ideas from the models built on living DNA. Yes, there was a major expansion of humans out of Africa, roughly 50–70,000 years ago. Yes, those people mixed with Neanderthals. Yes, later, waves of migration, like the spread of early farmers from the Near East into Europe, reshaped the genetic landscape. But they also reveal surprises that the models had blurred. Sometimes an ancient genome shows that what we thought was a single clean migration was actually multiple pulses over thousands of years, each with slightly different ancestry. Sometimes a burial in a region where modern people look genetically homogeneous turns out to belong to someone with a very different profile, evidence of a population that has since vanished or been absorbed. Each ancient individual becomes a kind of anchor point. Instead of inferring everything from the endpoints, us, today, you can fix intermediate dots along the timeline. With each dot, you can ask, are our previous models still the simplest explanation, or do they need to bend? When the first ancient African genomes began to appear, that bending became even more dramatic. A 4,500-year-old man from Mota Cave in Ethiopia confirmed that some East African groups have deep continuity in the region, while also showing that later back migration from Eurasia had altered nearby populations. 7 15,000-year-old hunter-gatherers from Malawi revealed a genetic profile not quite like any group alive today, suggesting that some ancient African lineages have no direct, unmixed descendants. Genomes from Stone Age individuals in South Africa pointed to an even deeper split among African populations than many models based on modern DNA alone had proposed. In other words, Africa's story was always central, but ancient DNA is showing that it's also more internally complex than any single Pan-African origin model captured. Sometimes an ancient genome sharpens an idea that was already there. It pins down the timing of an out-of-Africa movement, or the amount of archaic admixture with far more precision. Sometimes it complicates the picture, revealing ghost populations that contributed to modern groups but no longer exist as separate entities, or showing that lineages we lump together today were, in the past, more distinct. Both outcomes are progress. The rise of ancient DNA hasn't overturned the core conclusion that our species arose in Africa and later spread outward. If anything, it has reinforced that view by providing independent, time-stamped checkpoints. What it has done is transform that simple arrow on a map into something more like a braided river. Channels splitting and rejoining, side streams feeding in, some branches drying up while others grow. The more old bones we sequence, the more that river comes into focus. Not as a straight line from primitive to modern, not as neat boxes of isolated races, but as a shifting network of populations talking to each other through genes across space and time. Your DNA still remembers Africa, but it remembers much more than a single departure. It remembers detours and returns, encounters with other hominins, lost branches and hidden admixtures. Ancient DNA is the tool that's letting us hear those quieter harmonies, the ones that living genomes alone could only hint at. The story of origins is becoming sharper, yes, but with every new genome it also gains depth, texture, ambiguity. We are learning not just where we came from, but how tangled and interconnected that journey has always been. When people first heard that modern humans carry Neanderthal DNA, the reaction was often a mix of fascination and discomfort. What does that even mean? Are some of us part Neanderthal? Are Neanderthals our ancestors, our cousins, or something else entirely? To answer that, we have to rewind to a time after some of our ancestors had already walked out of Africa. Picture those early Homo sapiens spreading into the Levant, into Eurasia, into landscapes that were not empty. For hundreds of thousands of years other humans had been living there, Neanderthals in Europe and Western Asia, Denisovans further east, and perhaps other lineages we haven't yet named because we've only glimpsed them in fragments of DNA. We didn't replace a void. We walked into a neighborhood, Neanderthals first. For a long time they were drawn as brutes, hunched, dim, barely human. Ancient DNA tore that caricature apart. Their genomes show they were close kin, not some separate branch of life, but another twig on the human tree, split from our lineage maybe 500, 700,000 years ago. When scientists compared Neanderthal genomes to those of living people today, a clear pattern emerged. Individuals with ancestry from outside Africa, Europeans, South Asians, East Asians, Native Americans, consistently carried small chunks of Neanderthal DNA scattered throughout their genomes, usually around 1 to 2%, sometimes a bit more. Africans, in contrast, carried much less of this specific Eurasian Neanderthal signal. This mismatch is not random. It encodes an event, or more likely a series of events. Sometime after a group of Homo sapiens left Africa, perhaps around 50,000–60,000 years ago, they encountered Neanderthals and had children together. Those children were fully fertile. Their descendants spread, and over generations most of the Neanderthal genome was diluted away, but not all of it. A thin dusting remained, preserved in the stretches that happened to be neutral or even beneficial. Some of those Neanderthal variants affect your biology today. Certain immune system genes show a strong Neanderthal contribution, likely helping early Eurasians cope with unfamiliar pathogens. Some skin and hair variants trace back to Neanderthals. Even aspects of how you respond to ultraviolet light or metabolize fats can carry their signature. But 2% is not a second ancestry taking over. It's more like a faint watermark, a record of contact layered on top of a deeper foundation. Then there are the Denisovans. Unlike Neanderthals, Denisovans were almost unknown from bones. For years, all we had was that finger bone from Denisova Cave and a few teeth. Morphologically, they were a mystery. Genetically, they were a bombshell. Their DNA was clearly closer to Neanderthals than to us, but distinct enough to count as a separate branch. When researchers looked for Denisovan-like segments in modern humans, they found something striking. Most Europeans had essentially none. East Asians had small amounts, but some populations in Southeast Asia and Oceania, particularly Papuans, many indigenous Australians, and certain Philippine groups carried substantial Denisovan ancestry, on the order of 4–6%, sometimes even more when you add up different Denisovan-like pulses. That means at least one population of Homo sapiens moving through Asia into the Pacific met Denisovans and, again, had children with them, possibly more than once in different places, with Denisovan groups that were themselves regionally distinct. The oceanic record of that encounter is still written, very clearly, in the genomes of people living there today. So if you have ancestry from Europe or Asia or the Americas, parts of your genome likely come from Neanderthals, and if your roots lie in places like Papua New Guinea or Australia, some of your DNA also descends from Denisovans. The percentages are small but not trivial. They've shaped traits from altitude tolerance in Tibet to immune defenses in many populations. Here is where misunderstandings creep in. None of this means that Neanderthals or Denisovans are the ultimate source of Europeanness or Asianness, or that some people are less African because they carry archaic Eurasian DNA. The key is timing. Those interbreeding events happened after a group of Homo sapiens had already evolved in Africa, after they had become recognizably us in terms of overall anatomy and core genome structure, and after some of them had already begun dispersing out of the continent. Neanderthals and Denisovans were additions, side branches that rejoined the main trunk briefly, not the roots of the tree. Your genome, whatever mix of ancestries it contains, is overwhelmingly derived from that African-originating Homo sapiens population. The Neanderthal and Denisovan contributions are thin layers of varnish on an African-carved sculpture, and even the picture of Africans have no Neanderthal DNA has turned out to be too simple. Later back migrations from Eurasia into Africa carried small amounts of Neanderthal ancestry with them. Over thousands of years, as populations moved, mixed and remixed, some of that Eurasian-derived Neanderthal signal seeped back into African genomes. So today, some African groups carry detectable Neanderthal-like segments, but they acquired them indirectly via returning Homo sapiens, not from direct encounters with Neanderthals on African soil. More intriguingly, when researchers examine African genetic diversity in detail, they see hints of something else, archaic admixture that doesn't match Neanderthals or Denisovans at all. Think of segments of DNA in certain West or Central African groups that are unusually divergent, that branch off the human family tree much earlier than expected, if everything simply descended from one homogeneous Homo sapiens population. Those segments behave as if they came from populations that split from our line, perhaps several 100,000 years ago, and then, much later, interbred with ancestors of some modern Africans. We don't yet know exactly who those archaic Africans were. The fossil record is sparse, the ancient DNA record even more so, and multiple models can sometimes explain the same patterns. Some scientists propose ghost lineages, archaic human groups that lived in Africa alongside early Homo sapiens, contributed genes, and then disappeared as distinct entities. Others suggest long structured populations within early Homo sapiens that were partly isolated and then reconnected. Whichever model ends up closest to the truth, the implication is the same. Africa was not a single, uniform cradle. It was a continent-sized network of populations, some quite divergent, which occasionally exchanged genes. Eurasia, too, was such a network where Homo sapiens, Neanderthals, Denisovans, and perhaps others met and mixed. This is what admixture really tells us. Human history is not a tidy branching tree, where each split is final. It's a tree whose branches sometimes grow back together, a braided river rather than a single arrow across a map. Genes flowed between lineages. Populations split, drifted apart, then encountered each other again. Sometimes they merged almost completely, sometimes they left only a faint echo. Neanderthal DNA in a modern European, Denisovan DNA in a Papuan, archaic African signals in a West African genome. These are all variations on the same theme: contact, connection, reconnection, and through all those braids, the deepest channel runs back into Africa. Mixed ancestry after leaving the continent doesn't overwrite that origin any more than a few borrowed words from another language erase your mother tongue. It adds layers, complications, harmonies atop the underlying melody. In your DNA, the memory of Africa coexists with the memory of these encounters, with Neanderthals in Ice Age Europe, with Denisovans in Asian mountains and island chains, with shadowy archaic cousins within Africa itself. The story is not pure or linear. It was never going to be. And that complexity is not a problem to be solved. It's the very thing that makes our history human. So where does that leave the little pie chart on your phone that says you're 27% West African, 14% Scandinavian, 3% Indigenous Americas, and so on? It feels like the genetic version of a family tree, precise, authoritative, color-coded. But those numbers and categories live in a very different world from the one we've just walked through with Neanderthals, ghost lineages, and braided rivers of ancestry. To see why, you have to separate two things that often get blurred together: population-level research and consumer ancestry reports. When scientists talk about African origins, about Neanderthal admixture, about Denisovan signals in Oceania, they're working at the level of populations, not individuals. They compare many genomes across many groups. They build models, test alternatives, quantify uncertainty, argue over parameters. The result is not you are 1.9% Neanderthal in some personal sense. It's people with ancestry from this region tend to share this pattern of segments that look Neanderthal-like. It's a statistical landscape. Consumer tests take pieces of that landscape and turn them into a personalized weather report. They look at your DNA and ask, which modern reference groups do your genetic variants most closely resemble? Then they convert that resemblance into percentages and regional labels you can read in a few seconds. There's nothing inherently wrong with that, but it's crucial to understand how those pictures are drawn. Every ancestry estimate depends on a reference panel, a collection of DNA samples from people whose family histories are, as far as the company can tell, rooted in particular regions or communities. West African becomes the average genetic signature of the people they've sampled from places like Nigeria, Ghana, Sierra Leone. Scandinavian becomes the average of Norwegians, Swedes, Danes in their database. Indigenous American, Ashkenazi Jewish, East Asian, Southern Indian, Somali, Yoruba, Irish. Each label has a set of reference genomes behind it. Your results are essentially a measure of similarity. How much of your genome looks most like the variation patterns in each of those panels? Change the panel, and you change the comparison. Add more people from underrepresented regions, and you sharpen or sometimes completely redraw the borders. This is why ancestry reports change over time, even if your DNA does not. A few years ago, someone might have been told they were 20% broadly East African. Then the company sequences tens of thousands more people from Ethiopia, Kenya, Tanzania, Somalia, Sudan, updates its algorithms, and suddenly that same person's breakdown shifts to 5% Ethiopian, Eritrean, 9% Kenyan, 6% broadly East African. The underlying ancestry hasn't moved an inch. The map used to interpret it has been redrawn with finer lines. Think about that for a moment. The numbers feel absolute. 9%, 23%, 41%, but they're really coordinates on a moving map. As the reference data change, your reported ethnicity can slide from one region to another, or split, or merge. What looked like Native American 2 algorithm versions ago might now be divided into Andean and Mesoamerican. A chunk of Middle Eastern might later be relabeled as North African once enough North African genomes are added. This doesn't mean the tests are fake. It means they are estimates, built on comparisons, bounded by what's currently in the database. They are snapshots of how your genome fits into a particular company's model of human diversity at this moment, not timeless measures of your essence. There's another layer of confusion. The labels themselves, West African, Irish, Bantu, Italian, Native American, mixed geography, language, culture and history. But DNA is indifferent to our categories. It doesn't come stamped with flags or passports. The same genetic variant can be common in multiple populations for reasons that have nothing to do with how we draw political borders or ethnic identities. So when a report tells you you're 12% Nigerian, it does not mean that exactly 12% of your ancestors lived inside the modern boundaries of Nigeria. It means that about 12% of your genome most closely matches the set of people that company grouped as Nigerian, which might itself be a mix of several ethnic groups, each with their own history of migration and mixing. And when a report tells you you're 0% African, that does not mean you have no African ancestry in the deeper sense we've been talking about. It means simply that, in the time window, these tests are designed to detect roughly the last few hundred to maybe a couple thousand years. Your genome doesn't strongly resemble the particular African reference panels in their database more than it resembles others, but go back tens of thousands of years, and the story changes. Your maternal line, your paternal line, and the vast bulk of your genome all trace back, ultimately to African populations. That deep origin is shared, and it does not disappear because your recent ancestors happen to live in, say, Poland, Peru, or Pakistan. Consumer tests are tuned to detect relatively recent structure in human populations, the echoes of migrations, empires, enslavement, colonization, regional isolation, and cultural boundaries. They are not tools for pinpointing your exact village 50,000 years ago, or quantifying how African you are in some primordial sense. They can't tell you whether your specific lineage passed through a particular archaic African group, or how many Neanderthal encounters lie in your family's deep past. Those are questions we approach by aggregating many genomes and building population histories, not by reading a single pie chart. This is where marketing can blur into myth. Companies like to promise that your DNA will reveal who you are, unlock your story, or trace your roots back to the beginning. The language is powerful, almost spiritual. It suggests that the colourful segments on your screen are not just probabilities, but pieces of identity, heritage, even belonging. Used carefully, those reports can be meaningful. A person whose records were erased by slavery may find long-lost relatives or reconnect with specific regions in West or Central Africa. An adoptee may gain clues about biological kin. Diasporas can glimpse patterns of movement inscribed in their families' genomes. The danger comes when we mistake estimates for essences. If you start to believe that being 60% this and 40% that defines your authenticity, you're importing into your genome ideas that belong to politics, to culture, to the history of race, not to the biology itself. If you treat 3% here or 7% there as proof of a pure Viking soul or a trace of some ancient lost tribe, you're reading ancient identity into numbers that are, at best, a rough guide to where some of your forebears probably lived in the last few dozen generations. And if you use the absence of an African label in your recent ancestry report to deny the African roots of humanity, you're confusing two very different questions: where your great-great-grandparents lived and where our species became what it is. So when you hear scientific claims about human origins, Neanderthal admixture, or ancient African diversity, be wary of explanations that sound too much like a sales pitch. Real population genetics is messier, more cautious, full of margins of error and competing models. It does not divide us into neat, timeless ethnicities with hard borders. It talks instead about gradients, clines, admixture events, effective population sizes. Your consumer test can be a doorway into that deeper story, a way to spark curiosity about migrations and histories you never knew your family was part of. But it is not the story itself. To reach that, you have to look past the polished pie chart and back into the tangled, beautiful, African-anchored web of ancestry that all of us, without exception, carry. There's another trap waiting once you start staring at those pie charts and origin stories: what people do with them. Because information about DNA and ancestry doesn't stay in the lab or on your phone. It leaks out into politics, into identity, into old stories about us and them that have been told in a dozen different ways for thousands of years. The language of genetics can be borrowed, twisted, weaponized. Sometimes it's obvious. Claims that science proves one race is more intelligent, more evolved, more civilized. Claims that certain peoples are pure, others mixed, as if admixture were a stain instead of the universal human condition. References to European genes or African blood, as if they were discrete substances you can pour into a beaker and measure for worth. Sometimes it's subtler. National myths that lean on genetic studies to argue that a modern population is the direct, unbroken continuation of some glorious ancient group, the real descendants of the pharaohs, the true heirs of the Romans, the original people of this land as opposed to those newcomers. Or the reverse, stories of degeneracy, decline, replacement, framed as a dilution of some supposed original stock. The irony is that the actual genetics we've been talking about undermines these narratives at almost every turn. Human populations are not neatly separated, sealed-off branches on a tree. They're more like overlapping clouds, or colours blending into one another along a spectrum. You can detect clusters, yes. People from Japan, Nigeria, Finland, Papua New Guinea do not look genetically identical. There are patterns, there are gradients, there are echoes of geography, history, and isolation. But the differences are shallow compared to the shared depths. Genetic variation within any so-called race is often as large as, or larger than, the variation between races. Two people from the same village in West Africa can be more genetically different from each other than either is from someone in Europe or Asia. The boundaries we draw on maps, black, white, Asian, native, do not carve nature at some fundamental biological joint. They carve history, power, and perception. So when someone reaches for DNA to justify rigid biological race categories, they're importing an old social idea into a new scientific language. Geneticists do talk about populations, ancestry components, clusters. But these are tools for describing patterns, not declarations of essence. A West Eurasian component doesn't mean you have a Western Eurasian soul. A Bantu cluster doesn't fix your identity in place. These are statistical signals reflecting who had children with whom, under what circumstances over many generations. They're history written in code, not destiny written in stone. This is an important point, because the pendulum can swing too far the other way as well. Faced with the ugly history of racial science, eugenics, craniometry, Nazi ideology, there's a temptation to say race is entirely made up. Genetics shows we're all the same, end of story. But that's not quite right either. We really do see regional adaptations in human DNA. Darker skin near the equator as protection against UV radiation. Lighter skin in higher latitudes to help synthesize vitamin D. Variants that help digest lactose in populations with a long history of dairying. High altitude adaptations in Tibetans and Andeans. Malaria resistance in parts of Africa, the Mediterranean, South Asia. These are real differences. They have real consequences for health, for physiology, sometimes even for how drugs are metabolized or diseases manifest. And culture, too, is not an illusion. Languages, religions, cuisines, kinship systems, political structures, they shape lives just as powerfully as genes do, often more. A shared history of oppression or migration can create bonds that no DNA test can measure. The fact that all humans share African origins does not erase the difference between a Yoruba farmer, a Gujarati shopkeeper, a Sami reindeer herder, and a Quechua musician. Deep common ancestry and real diversity are not opposites. They are layers in the same story. The risk is when we try to flatten those layers into a single axis, more or less X, higher or lower Y, as if one number, one category, one gene, could sum up a person's worth or fate. That's where a new myth begins to creep in, the myth of genetic destiny. You can see it in headlines about a warrior gene, a cheating gene, risk-taking DNA. In glossy promises that your genome can reveal your ideal diet, your perfect workout, your innate talents. In attempts to link complex traits, intelligence, aggression, musical ability to simple genetic markers, then map those markers onto populations. But the traits that most obsess us, behavior, cognition, personality, are influenced by thousands of genetic variants, each with tiny effects interacting with each other and with environments we barely understand. Education, nutrition, pollution, trauma, wealth, discrimination, family structure, language, all of these shape outcomes just as powerfully as whatever variants happen to sit on chromosome 3 or 11 or 22. Genes set possibilities. They bias the deck. They do not play the cards for you. So if the old myth said, humans are divided into a few natural races, fixed and hierarchical, we shouldn't replace it with, your DNA sequence is your future, your identity, your truth. That's just swapping one kind of determinism for another. The more we learn, the more the story resists simple slogans. We are a young species with deep African roots, shaped by migrations, mixtures, and local adaptations. We carry hints of Neanderthals and Denisovans, echoes of ancient African lineages, signatures of famine and plague and empire and enslavement. We are neither uniform marbles in a jar nor isolated, unmixed essences in separate boxes. That complexity puts a responsibility on our words. When we say African origin, we're not saying everyone is basically the same, or Africa owns you, or your recent ancestry doesn't matter. We're saying the earliest chapters of our species' story were written there, and traces of that origin are still detectable in every genome on Earth. When we talk about European or Asian or Native American ancestry in genetics, we're not endorsing some timeless natural division of humanity. We're using imperfect labels to point at statistical patterns that happen to align, roughly, with regions and histories. When we talk about black or white in medical studies, we're often using social categories as proxies for a messy mix of genetics, environment, and lived experience, a practice many researchers are actively trying to refine and improve. Precision matters, not because pedantry is fun, but because sloppy language creates openings for old ideas to sneak back in wearing new clothes. So as you think about your own ancestry, about that African beginning, about the threads that connect you to people scattered across the globe. It helps to hold two truths at once. You are not reducible to your DNA. You are not a percentage breakdown or a reference panel match. Your identity, your belonging, your culture are built from stories, choices, relationships, histories that no sequencer can read. And yet in every cell there is a record that you are part of a single sprawling, entangled family that began its strange experiment somewhere in Africa hundreds of thousands of years ago. Understanding that record clearly without myth, without ideology, without oversimplification, is what lets us use it wisely. And it's what we'll need as we turn to the final question. Given all this complexity, all this mixing, all these caveats, what counts as the strongest proof that humanity's story begins in Africa? And what do we still not know? Let's start by pulling the threads together. Throughout this journey, you've heard about fossils, about genetic diversity, about mitochondrial DNA and Y chromosomes, about ancient bones in caves and modern people spitting into tubes. What, taken together, rises above the noise as the strongest evidence that our species has deep African origins? First, the fossils. When you line up the known remains of creatures that look recognizably like us, rounded skulls, relatively small faces, chins beginning to appear, the earliest and most modern in form keep clustering in Africa. Jebel Irhoud in Morocco. About 300,000 years old. Homo Kibish in Ethiopia, around 195,000 years. Herto in Ethiopia, roughly 160,000 years. These skulls aren't identical to a modern human, but they're close enough that if you passed one of their owners on the street today, you'd probably just think, that's a person outside Africa. Fossils with clearly modern features arrive later. Skul and Kafse in Israel, around 100,000, 120,000 years ago. Sites in Greece and China that suggest brief early ventures out of Africa. Then a more persistent presence in the Levant, Europe, and Asia after about 70,000 years ago. The pattern is simple, even if the details are messy. Modern-looking humans show up first in Africa, and only afterwards elsewhere. That's one pillar. The second pillar is genetic diversity. If you measure how different two random people are at the DNA level, how many little changes they have between their genomes, you get a surprising result. Two people from sub-Saharan Africa are, on average, more different from each other than either is from a person in Europe, Asia, or the Americas. In other words, Africa holds the deepest reservoir of human genetic variation. This is exactly what you'd expect if our species had been living in Africa for a long time, accumulating mutations in many semi-connected populations, and then only a small subset of those populations later left the continent. That subset effect is called a bottleneck. It leaves a signature, lower diversity, more genetic uniformity, in the descendant group. We see that bottleneck in everyone whose ancestry traces outside Africa. We do not see it to the same extent within Africa. Instead, we see layers of older structure and variation that must have been building up long before those outward migrations. The third pillar comes from multiple independent genetic systems, all telling the same broad story. Mitochondrial DNA, inherited from mothers, coalesces to a common ancestor, the famous mitochondrial Eve, who almost certainly lived in Africa about 150,000 to 200,000 years ago. The Y chromosome, passed from fathers to sons, traces back to a Y-Chromosomal Adam, who also lived in Africa, perhaps between 200,000 and 300,000 years ago, depending on the estimates. Autosomal DNA, the bulk of your genome that recombines every generation, points to the highest ancestral diversity and deepest lineages inside Africa, with all non-African populations branching off from within that African variation. And then ancient DNA from Neanderthals and Denisovans adds a fourth pillar by exclusion. Modern non-Africans carry bits of Neanderthal and Denisovan ancestry. Most sub-Saharan Africans do not. except where there has been recent back migration, that implies that the mixing with these archaic Eurasian humans happened after a group of modern humans left Africa, met them, and had children. 4 independent lines, fossils, diversity patterns, uniparental markers, and archaic DNA are all converging on the same broad picture. Our species arose in Africa, diversified there, and only later spread across the rest of the world. That's the part of the story that is, at this point, extremely hard to escape. To overturn it, you'd need not just a surprising new find, but a complete rewiring of how evolution, population genetics, and the fossil record fit together. But outside that core, the ground quickly becomes less solid. We know modern humans originated in Africa. We don't yet agree on exactly where in Africa, or even whether there was a single where. One major open question is internal African population structure. Did Homo sapiens evolve in one primary region, a sort of cradle somewhere in East or Southern Africa from which everyone else descends? Or did our species emerge from a network of semi-isolated populations scattered across the continent, connected by occasional gene flow, gradually converging on the modern human pattern? This is sometimes called the Pan-African or metapopulation model. It would mean that when we say African origins, we're really talking about a continent-wide process, not a single Eden. Current data lean towards some form of that network idea, but the details, how many groups, how separated, how often they mixed, are very much under debate. Then there are the migration routes and timings. We know there were early excursions of modern humans out of Africa. Some fossils in Israel, Greece, maybe China, that likely did not contribute much, if anything, to the DNA of people living today. We also know there was a major successful expansion, roughly 50,000, 70,000 years ago, that did leave lasting genetic traces across Eurasia, Oceania, and eventually the Americas. But the precise paths are still fuzzy. Did the main wave hug the Nile and the Levantine corridor, or cross the Bab el-Mandeb Strait into Arabia and spread along the coasts? Were there multiple waves that later blended, rather than a single clean exodus? How much of today's non-African ancestry comes from each? Different models using different data sets and assumptions give different answers. None of them is nailed down the way the broad African origin is. Even within Africa, the timing and sequence of splits between ancestral populations remain a tangle. When did the ancestors of West Africans diverge from those of East Africans? How do hunter-gatherer groups like the San and the Hadza fit into the tree? When did deeply divergent ghost lineages, ancient African populations we only glimpse through their genetic echoes, branch off and disappear? These are the kinds of questions where small changes in assumptions about mutation rates, population sizes, or migration events can shift the answer by tens of thousands of years. And then there's the great unknown, ancient DNA from Africa itself. Most of the ancient genomes we currently have come from Europe, Siberia, and a few parts of Asia and the Americas. That's not because those places are more important, but because their climates are kinder to DNA. Cold, dry environments preserve genetic material, hot, wet ones destroy it. Africa, especially tropical Africa, is brutal on ancient DNA. Bones decay faster. Molecules break apart. Until recently, people weren't sure we'd ever recover very old genomes from most of the continent. That's starting to change. Over the last few years, teams have managed to sequence DNA from African individuals who lived tens of thousands of years ago, from Malawi, from Ethiopia, from Morocco. Each new genome has forced revisions to our models, revealing unexpected mixtures, population turnovers, and connections that weren't obvious from modern DNA alone. And that's just the beginning. As techniques improve, better extraction methods, better contamination control, better sequencing of tiny fragments, we're likely to get more windows into the African past. Every one of those windows has the potential to reshape parts of the story. They might show that some lineages we thought were extinct actually survived longer. They might reveal previously unknown groups that contributed to modern populations in ways we haven't guessed. They might tighten the dates for key splits or migrations, or overturn neat trees in favour of messy webs. So where does that leave us? On solid ground for some claims, and on shifting sands for others. Well supported that Homo sapiens emerged in Africa, that African populations hold the deepest genetic diversity, that all non-Africans descend from a subset of Africans who left within the last 100,000 years or so, that we met and mixed with other humans like Neanderthals and Denisovans along the way, debated the precise geography of our origin within Africa, whether one region or many interconnected regions better describes it, the exact routes and number of successful out-of-Africa migrations, the detailed branching order of African ancestral populations, the contributions of now extinct ghost groups inside Africa, uncertain but promising, how much African ancient DNA will ultimately be recoverable, and how radically it will redraw the finer branches of the tree. So as we move to the final part of this story, it's worth holding on to that distinction. There is a strong, robust core. Your DNA really does carry an African memory, and that memory is not a metaphor. It's built from mutations, recombination events, and population histories that we can measure, model, and cross-check against bones and stones. Around that core is a halo of questions, some of which may be answered in your lifetime, some of which may always remain a little blurry. The goal is not to inflate the certainty of what we know, or to pretend the open questions don't exist. It's to be precise about both, to see the outline clearly, and to be honest about the unfinished lines. Because the value of this story isn't that it gives you a simple, triumphant, one-sentence origin myth. It's that it roots you in a real, evolving, testable understanding of where you come from, and what it means and doesn't mean to say that your DNA still remembers Africa. So what do you do with all of this? It's one thing to know, abstractly, that your genome has a long trail of mutations that lead back through time, through bottlenecks and expansions, through deserts crossed and coasts followed. It's another thing to sit with the implication. Every living human, everywhere on Earth, is stitched into that same story. Your DNA is not a flag. It isn't a purity test or a ranking system. It's a record of movement and mixture, a record of populations splitting, wandering, meeting again, having children. It's the story of people who kept leaving home and then redefining what home meant. When geneticists say African origins, they're not saying everyone today is the same, or that culture and history and place don't matter. They're saying that if you keep following the branches of our family tree backwards, if you keep peeling away the more recent layers of migration and empire and nation, you reach a deep trunk that is anchored in Africa. That trunk is real. It's supported by fossils, by patterns of diversity, by mitochondrial lineages and Y-chromosomes, by the scars of ancient interbreeding. Your DNA really does preserve strong, measurable evidence that our species emerged and diversified in Africa before radiating across the globe. And that matters because it cuts against some very old stories that tried to do the opposite, to divide, to rank, to invent separate creations. The genetic evidence doesn't erase difference, but it puts all of that difference inside a shared ancestry. It makes racism, in a literal biological sense, incoherent. The categories we use to separate us and them float on top of a common origin. It also matters for how you see yourself in time. You are not just the product of your recent family or your passport. You're the current endpoint of a 4 billion year experiment in life, and a few 100,000-year experiment in being human. The migrations you see on the news, the debates about borders and belonging, are just the latest chapter in a story that began with small African populations edging into new valleys, new climates, new risks. Remembering that doesn't solve today's problems, but it can shift the frame. It turns those people over there into distant cousins whose ancestors walked the same rift valley, under the same sun, facing the same uncertainty. So if this journey through DNA and deep time gave you something to think about, if it nudged the way you see yourself or other people, take a second to let me know. Tap like if you'd like to see more long-form dives into human origins and genetics. Subscribe if you want to be there for the next chapter in this unfolding story, as new fossils are found and new genomes are sequenced and our picture sharpens. And I'd love to hear from you in the comments. Where are you listening from right now? What part of the world are you in, adding your own small modern data point to this very ancient map? Drop your location, share your thoughts, and I'll see you in the next one.